Hello, this is Richard Jacobs with the Finding Genius podcast. I have a returning guest, Dr. Thomas Seyfried. He's a professor at Boston College. He's part of the biology department, and he focuses quite a bit on cancer. He's written several books about it, and I'm having him here for a new cancer publication book I'm putting together where I'm going to interview, several dozen cancer researchers. So, Tom, thanks for coming. I appreciate you being here.
Oh, thanks, Richard. It's nice to be here.
Yeah, just to begin, could you just give a background on what's your current research in regards to cancer about right now?
Well, our current research right now is to develop a diet drug cocktail that can be used to provide non-toxic long-term management or possible resolution of the disease in its entirety.
Okay. Do you tend to focus on primary tumors or particular cancers or metastases, or like what? Within cancer, there's a lot of areas. So what are you focused more on the metabolism of cancer? Or?
Well, you have to realize that all cancers are the same, whether it's brain, colon, breast. They all have the same underlying problem. They can't use oxygen to generate energy, so they ferment. There are only two major fuels that drive fermentation, and that's glucose and glutamine. So if we can effectively target and restrict glucose and glutamine with diet drug combinations, every known major cancer should be potentially manageable.
Yeah, that would be phenomenal. You said they can't use, from what I understand, cells do what's called oxidative phosphorylation to harness ATP. And then when you speak about fermentation, what's that process called and how is it different from oxphos?
Well, fermentation is an ancient pathway. It generates energy without oxygen. And all organisms that existed on the planet before oxygen came into the atmosphere some 2.5 billion years ago, the organisms at that time were all fermenters. There was no oxygen. So they would reproduce unbridled reproduction driven by fermentation metabolism. It's a very efficient way to get to generate, to reproduce, proliferate, and keep the energy requirements in the cell stabilized. Now, of course, when these bacteria were able to capture and use oxygen to generate energy, that became the main source of development of metazoans, let's put it that way, the multicellular organisms, which gave them the opportunity to do a lot more, perform a lot more functions and sophistications with a more efficient source of energy coming from oxygen as a final acceptor of electrons. So this opened up the whole vast evolution of organisms on the planet. But those ancient pathways of fermentation are still present in every one of the cells of our body. The glycolytic pathway is an ancient metabolic pathway. And so is the glutaminolysis, amino acid fermentation. But these are ancient pathways. It's just they're very, very minor in most of the cells of the body of metazoans, all mammals. And so we're falling back. The cancer cells simply fall back on these ancient pathways to survive. And once you know that, the biology is overwhelmingly consistent with this. Once you know this, then you know how to target and manage the disease.
So what are some of the trade-offs of oxphos versus fermentation? Is it less efficient? What's good and what's bad about it, this pathway?
Well, fermentation can keep cells alive for a short period of time. If you hold your breath long enough, your face will start turning blue. The idea here is that, but you're not going to drop dead instantly because our cells are going to be upregulating massive amounts of glucose into the cells for glycolytic metabolism to get ATP through glycolysis. But that can only last for a short period of time. It can't sustain you. The best evidence, you have a heart attack, you don't live long. The neurons in our brain are very, very sensitive to deprivation of oxygen. So we essentially die from brain damage from heart attacks. But we don't die instantly. We have some. That's why you can rescue people. But it's a very inefficient process because the blood fills up with lactic acid very quickly. That's why you're turning purple. You can't get the energy. You can't do this. But it can't sustain the high energy demand of the cells of our body. The fermentation can only help us for a short period of time. Then if we don't get oxygen, we're going to be dead. So, when you want to kill people fast, you drink the cyanide, right? They call it taking the Kool-Aid. Well, it was filled with cyanide. Cyanide shuts down oxidative phosphorylation almost instantly, right? People die from cyanide very quickly. Cancer cells live in cyanide. So clearly they're not using oxygen. So tumor cells live in cyanide. They can grow fine in cyanide. that tells us that they're fermenting. All right, they're using fermentation. They're using glucose and glutamine to stay alive in the presence of a complete shutdown of oxidative phosphorylation. So all you have to do is target the glucose and glutamine and you'll kill these guys. It's not that complicated.
Well, I mean, from what I understand, it is possible to have a diet that's incredibly low in glucose, but glutamine, I believe, is used in many cellular processes. How can you create an environment of low glutamine for a period of time?
Well, you have to use drugs and you pulse them. You don't target glutamine. You're absolutely right. Glutamine is essential for the urea cycle, essential for our immune system, essential for our gut health. Glutamine is an extremely valuable amino acid. So if you're going to target glutamine, you have to be very strategic in doing it. Because if you do it too aggressively and you don't understand the biology of the problem, you can do significant harm to the gut, to the immune system. So and another thing, if we kill cancer cells in large numbers, it's the immune system that comes in and cleans up the dead cells in the microenvironment. If you use glutamine the wrong way, those dead cells persist in the microenvironment, leading to all kinds of adverse consequences. And so you have to be very strategic about it. You got to know what you're doing. It's not that it's so complicated. It's just that you have to be knowledgeable and educated to understand how to do this. And this is what we're doing. And once we're successful in doing this, most people will say, wow, I can't believe it could be that simple. So the problem is most people, what I just told you is not known to the majority of people.
So from your studies, how do you believe cancer first starts? What will happen in cells first and then the next and next and next?
Well, as Otto Warburg clearly said, it's a chronic interruption of oxidative phosphorylation over time. So, I mean, you are not a perfectly healthy person one day and wake up with a massive tumor on you the next day. These things happen gradually. So we know what causes cancer. It's damage to the oxidative phosphorylation system with a compensatory transition to fermentation. It's that transition to fermentation and the disruption of oxidative phosphorylation that leads to the genomic instability and somatic mutations that you see in the nucleus. So the mutations and genetic damage is all downstream epiphenomena. So we know what causes cancer. You know that carcinogen. Why they call a compound a carcinogen? I don't know. I'm not assuming that I know, because I've heard that some viruses can cause certain cancers, and the other theory seems to be just random mutation, which I don't agree with. Yeah, but where does the random mutation come from? Well, from what you're saying, if there's a stress, maybe there's active remodeling of the genes that causes what we perceive as a mutation, but maybe it's a stress response, and that's one of the facets of it. So what it is reactive oxygen species, and we know those are a lot of cancer cells. Almost all of them are throwing out reactive oxygen species. ROS are carcinogenic and mutagenic, so we can cause nuclear gene mutations through unregulated reactive oxygen. Reactive oxygen species are essential for cell signaling cascades, but when they get out of control, they can lead to a genomic destabilization and somatic mutations. There was a couple of beautiful papers showing that P53 defects lead to tremendous ROS, causing more somatic mutations. So the somatic mutations are really a downstream epiphenomenon.
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
Do you think then that, so OXPHOS goes wrong, and do you think there's an abundance of oxygen then in the local cell microenvironment, and some of that through, improper OXPHOS gets turned into radical oxygen species, which then mutate the genes?
Yeah, I think that's pretty accurate. This is a cascade of events. And like you said, the viruses, we looked into this, all of the oncogenic viruses like papillomavirus and hepatitis C and some of these other viruses, they enter normal cells, and either the viruses themselves or the product of the viral genome goes into the mitochondria, destabilizing oxidative phosphorylation. And this can happen, over time in a particular population of cells in the target organ, leading to unbridled proliferation in those cells. And when you look at the cells of those viral-induced tumors, they're all fermenting like crazy. But the mechanism is like, how did that virus in the first place lead to the formation of a bunch of cells that are dividing uncontrollably? And when you look back, the virus has targeted the oxidative phosphorylation system, leading to compensatory fermentation.
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the general scientific concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer.
So you can look at X-rays will cause cancer, and we know that X-rays damage OXPHOS. Intermittent inflammation, like you have intermittent hypoxia and inflammation. I mean, you in a milk duct of a breast or in a prostate or some other organ, if you get intermittent hypoxia or inflammation, you block and damage the respiration of those cells that are suffering, thereby leading to the initiation of a tumor. Age, of course. And then, of course, people always say, well, it's got to be genetic because you have BRCA1 and P53 leaf round many and these things. The interesting thing is that those mutations will only cause cancer if they damage mitochondria. And we know there are about 50%, maybe 40% of women that have the BRCA1 mutation never develop breast cancer because for whatever reason, which we don't know, the mutation in those individuals never damaged their mitochondrial respiration. So those are what we call risk factors. They're risk factors. They're not the cause, but they will increase your risk for cancer. We can go down the list and look at every one of the provocative agents that can cause cancer. And it was referred to by the Nobel laureate Albert Szent-Györgyi that as the oncogenic paradox. He said, well, geez, you can get cancer from almost everything. And we don't understand how we can get dysregulated cell growth from almost all these provocative agents. And all the provocative agents, in one way or another, damage respiration, leading to compensatory fermentation and the path to unbridled proliferation. Once you know how to connect the dots, it does not become that complicated.
So what do you think the first thing that happens to a cell is that its mitochondrial function is impaired? Like what's happening in the mitochondria, you think, that starts off this whole cascade?
Well, I mean, if we're exposed to a chemical carcinogen, the carcinogen enters into a particular cell. Now, of course, if the damage to the respiration is acute, in other words, it happens really quick, that cell will die. Just like if it were consuming cyanide, it dies, and you can never get a cancer from a dead cell. This was clear. It's the chronic interruption of respiratory function, and this could happen over decades, or let's put it this way, years. There are some cancers that can happen, interestingly enough, like within months of the insult, but they're not generally the common ones. The common ones like breast cancer, colon cancer, lung cancer, prostate cancer, these all arise as a protracted interruption of oxidative phosphorylation in a particular cell or population of cells in the tissue, which then leads them to throw out lactic acid, destabilizing the microenvironment, leading to further damage to cells in the surrounding neighborhood. And all of a sudden, you have a neoplasia. The interesting thing, though, is that our immune system recognizes this, and we have cells in our body that recognize these localized disturbances. Problem is, many of these cells are macrophages, and they come in to try to restabilize or put out the fire, and they throw out growth factors and cytokines. It's like putting gasoline on the fire. It's an inappropriate response to a particular situation. And that situation gets worse. And what happens is these macrophages then fuse with some of these localized, destabilized cells. And eventually, their mitochondria become corrupted through a dilution process in the cytoplasm. And now these cells are the origin of the metastatic cell. And metastasis is ultimately the process of cancer that kills most people. And you understand what metastasis is. You understand what the cell is involved with that process. And you understand that cell needs massive amounts of glutamine and glucose to do its job. And now you know how to control metastasis.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
All right, so question here. So when a cell becomes cancerous, you said it'll tend to release more lactic acid from its altered fermentation respiration?
Yes, lactic acid and succinic acid. And there's a destabilization of the microenvironment that also contributes to a facilitatory action to further destabilize oxidative phosphorylation. So it's kind of like a vicious circle. Eventually, you get a cell that now is destabilized.
Well, I've seen that many cell types and tissues in our body have their own microbiomes, skin microbiome, vaginal, gut, eye. Perhaps the entire body has, localized microbiome adjacent to any cell. So when the cell gives off more lactic and succinic acid, do you think that a disruption of the local microbiome for that cell is a huge factor in terms of, facilitating metastasis, or what do you think it does?
Well, I don't call it the microbiome. I call it the microenvironment. It's the tumor cell microenvironment.
But some of the players in the microenvironment of tumor cells, like for instance, I interviewed Florencia McAllister, and she said that they were studying pancreatic tumors, and they noticed that the tumors themselves had different localized microbes that hung out with them than the rest of the pancreas. So that's what I mean, like part of the microenvironment. Would you say one of the critical players is the local microbes, bacteria, protists, et cetera, that are there?
Yes.
How are they affected then?
We have evidence now that mycoplasma and certain other papillomaviruses and these other microbes are facilitators of fermentation metabolism. Can you believe this? So we know this. There's several papers published on this very process. In other words, the cancer cell will ferment as the result of its oxidative phosphorylation defect. But, as you just mentioned, these microbes that are infecting the cells, they're inside the cells themselves, are facilitators of enhanced fermentation. And, we study, many people in the research industry in cancer study all these highly purified cells and tissues and these germ-free mice. And when you take tumors out of people and you look at at what's going on, you see you're exactly right. You see all these internal microbes and all this kind of stuff. And they are facilitators of the fermentation. So they are pushing this cell further into destabilized energy metabolism.
Wait, so a damaged cell, wow. So there's microbes adjacent and outside the cell membrane, but you said there's also microbes literally in the cytoplasm of the cell?
Mycoplasma. Wow. Cytomegalovirus. They're inside the cells.
So that would mean that there's going to be a turnover. It's like a new administration will come in once the cell abandons OXPHOS and goes more to fermentation. Like, I guess you'd expect all the interior and exterior localized microbes to change over and to facilitate this new form of respiration.
Yeah, I think they're contributors. The bottom line is that it all comes back to, they're facilitating, but they're not, if you can they're still going to be driving the fermentation metabolism, which is ultimately what you're trying to target.
Well, if they're driving it, what about
I wouldn't say that. I mean, they're facilitating. They're contributors of this.
Contributors. Has it been identified which particular mycoplasma or bacteria or whatever it may be are the strongest facilitators of fermentation?
No, but there was a nice paper by Dr. Yu who evaluated these things quite thoroughly. And he was the one who, and others have shown that human cytomegalovirus, which is an infection of a number of cells, they can contribute to this.
If science could figure out which particular organisms were the most, that show up and are the most facilitating of this fermentation, and if you were able to dampen them, that might also be a good adjunct in the therapy, not just glucose and glutamine restriction, but this may be yet another way to help slow it down or stop it.
Yeah, well, they have some drugs, valcyclovir. It's a drug that targets some of these microbes to try to. They did this for glioblastoma out of Sweden, I believe. or Norway, in one of the Scandinavian countries. Valcyclovir, it's one of these antiviral medications. And they were able to slow down GBM a little bit. But, the bottom line is the cells are still using glucose and glutamine. So regardless, if you kill the cell by taking away its fermental fuels, you don't have to worry about the microbes. They're going to die with the cell. So, the bottom line is that what's the goal of all of this? The goal of all of this is to get rid of the cancer.
But you're hinting that glutamine restriction, you have to be real careful with it. So maybe that would be the reason maybe to incorporate these other methods too, because then it makes the glutamine restriction easier and less prone to, collateral damage. That's why I suggested.
Yeah, well, it might be. I'm certainly not going to exclude it. I mean, if that could be added as part of the cocktail, certainly.
Has anyone contemplated phage therapy? The microbes that are inside the cells or around the cells that facilitate the fermentation to stop them?
I don't know about that, but, it's much easier just to shut down the glucose and the glutamine. I mean, you can do a lot of things. When you develop a strategy, you have to say, what is the probability that I'm going to resolve the disease with this therapy? What is the probability that I'm not going to harm any of the functions of the normal cells and what they do? And how easy is it to do? So you put all these things together and you come to because there's a lot of Rube Goldbergs out there, and these are very highly sophisticated procedures to try to stop cancer, like CAR T immunotherapy and some of these PD-L1 and all this kinds of stuff. I mean, these are very complicated, sophisticated kinds of things that in my mind are largely unnecessary if I can get rid of the tumor in a much easier way without toxicity. So, I mean, yeah, you can use very expensive, very elaborate procedures to try to kill cancer cells, and this is what we're primarily doing in the clinics today. But, I think there's other ways that we can do the same. What is the goal here? The goal is to kill cancer cells without harming the body, right?
Yeah.
That's the ultimate goal. And I'm saying we have a plan to do that. And it's very clear, and it's based on hard science.
No, I agree with you. It sounds great. Are you able to get clinical trials going for this, or what stage is this this at for you?
So when you say clinical trial, it's a very costly procedure.
I know the reason I say it, though, is that, unfortunately, that seems to be what everyone wants is clinical trials.
Sure.
So,
Well, who doesn't? The idea here of a clinical trial is double-blind crossover, right? Well, if you're using diet and drug combinations and cocktail procedures, this kind of a strategy is not consistent with the double-blind crossover pharmaceutical model, okay? And then when you do a clinical trial, most of the times that this is supported by major pharmacies. And I haven't seen yet a pharmacy company step forward to say, okay, let's try diets with non-patentable drugs. This just is not part of the financial situation right now, right? It makes sense. So,
if it's going to include diet and drugs,
yeah,
maybe you have to chip away at it. And first thing you do is do clinical trials with some drugs.
Yeah.
And then maybe you add the diet in on a separate one or both. I don't know.
Well, I mean, if you're the one with the brain tumor, you have two choices. You can go or the advanced lung cancer or whatever. You can say, okay, let's just kind of chip away at this and let's see what we can do. But if it's your ass on the line, you'd be surprised how your viewpoint changes. And, the people that want to stay alive and understand the biology of the problem are eager to do this. We're working on a case report right now where a young man who chose for glioblastoma chose no radiation chemo, no standard of care, but metabolic therapy alone. Now he's out almost 7 years. He had documented glioblastoma, and he's managed it with non-toxic metabolic therapy. And I'm telling you, when this paper appears in the literature, it's going to make a lot of eyes wide open. Why do you take cancer patients and subject them to these horrifically toxic therapies? And you end up with the patient being compromised metabolically and physiologically everywhere. And oftentimes these poor folks die. And we don't have to have this. This is unnecessary. And these what I'm telling you now is not well received, and it's mostly ignored for obvious reasons.
I'm sure it is.
Yeah.
Well, what do you work with a clinic that is able to recommend some of these strategies? Or how do you get this hypothesis tested? How do you do it?
Yeah, well, what we do is we take individual patients and we put them on these therapies, and then we publish it, okay? And it goes out into the scientific literature. then the field can decide what they would like to do with this information. Do they want to?
How do you get individuals to do that? Is it if it's just one person and they choose to do it? Is that how it gets around? It's like considered self-experimentation, or like what?
Yeah, well,
what are the rules?
Yeah, it's not easy because you're driven by a medical establishment to say do this, and when you go to the top medical schools, they will tell you, oh, we need standard of care, which is radiation and chemo and whatever else that might be added to this. Then if that doesn't work, well, then maybe we'll consider a metabolic approach. Well, we've published a major paper with major physicians and people in the field showing how the standard of care actually contributes to the rapid tumor recurrence and death. So you're actually stacking the deck against survival by the very treatments that the establishment would like you to use. And, the death rate of cancer is still very, very high. And I'm not saying some of the newer therapies haven't worked. Of course, there are people that survive these with the newer therapies, but many people have died quickly, very rapidly from some of these new therapies, which is a tragedy in itself. But the bottom line is, so they're running big clinical trials right now at Cedars-Sinai on ketogenic diet metabolic therapy for brain cancer only after standard of care. So my argument is that if you did metabolic therapy without standard of care, what would the outcome be? And nobody wants to touch that with a 10-foot pole.
Yeah, I'm sure they'll cry that it's unethical, even though
You're talking about ethics? You're a young, healthy guy, right? Do you have any health issues right now?
Well, I've had thyroid cancer.
But right now you're feeling pretty good, right?
I feel good, but you never know,
okay.
yeah.
So if we took you down to Dana-Farber, MD Anderson, and gave you a treatment that they would give to advanced pancreatic cancer or brain cancer, I can guarantee you right now you would be one sick buckaroo.
Oh, I believe you. 100%.
Okay.
So, is there any chance that you could do it without standard of care?
Yeah, the guy we're working on right now took no standard of care. He's doing fine.
But how does that work? I would think, do people come to you before they go to the doctor, or if they're seeing a doctor, how do they work with you and then tell the doctor what's going on? And the doctor will be like, no, don't do that.
No, I never tell anybody anything like that. So if they come to me before, when they first diagnosed, I give them information, published articles in the scientific literature to educate them. Then they would go and talk to their oncologists or whatever, and then they would come to a group decision. So if the patient knows that if I go the course of standard of care, especially for glioblastoma, I have a 98% chance of not surviving for 3 years. All right. But if I do metabolic therapy, it's unknown what the outcome will be, but I could live as long or longer. And certainly my quality of life would be a hell of a lot better. So the patient and the family have to come to that decision. I don't tell them what to do or what not to do. I simply show them the scientific papers from the literature, and here's the outcome. And a lot of these poor people have never seen survival statistics from the very treatments they are given. The doctor doesn't come in and say, look, let me show you where you're going to fall on this survival chart. They don't know that. And many of these poor people think that the treatments they're getting are going to be curative, not palliative. Palliative is only to keep you alive a little bit longer. It's not to. But yet they view the treatment that they're getting from these top oncology centers as potentially curable. And that's misinformation. You got to look at, show the survival curve at your age, at your situation, where you think you're going to fall on your survival curve. And that opens your eyes real fast. What else is there? Doc, what else is there? And the answer is no, this is the only path you have. And that's misinformation. These guys don't know the stuff that I just told you. They need to read the scientific literature and look at, there's no evidence to support that. That might be true, but there's plenty of evidence.
So the people that you're working with, is it called anecdotal, or what would you call it? What how many people have you worked with and helped, and what do you call that bulk of information? Like, what do you do with it?
No, we published several papers, and the guy we had from Egypt who was really doing very well, a young man who had glioblastoma, his whole left side was paralyzed, and is a corn farmer. And they came to me, the oncology group asked if metabolic therapy would help this guy. So we actually put him on metabolic therapy for 3 weeks before we did the debulking surgery on this individual. He was doing great. It really, really came well. And then we postponed radiation therapy for 3 months, which is unheard of in the oncology field. But they pressured to give this poor guy massive doses of radiation. And I told the oncologist, I said, why are you doing this? The guy is really doing well. He's very healthy. Oh, no, we have to do this. Everybody in the world does this, which is true. So they irradiated this guy, and he did pretty good. He was doing very well. He survived the radiation treatment and the temozolomide and all this so-called standard of care. But then and we published the paper at 24 months. So he was surviving for 24 months with pretty good health. But then at 30 months of age, he started developing headaches, and he died very quickly. Autopsy showed massive radiation necrosis damages his brain. His brain was liquefied from the damn radiation.
Jesus.
So what the hell are you talking about? What you called it earlier? Is it ethical? You even said, is it ethical to go and do metabolic therapy over standard of care? I'm saying, is it ethical to irradiate somebody's brain until it liquefies?
Well, no, I agree with you. I brought it up because I'm sure that's what a lot of medical people would say. It's unethical to do that.
Yeah, well, it's unethical to irradiate somebody also.
I agree. I agree with you.
Let me clarify the situation. I'm not opposed to radiation therapy for every kind of a cancer. In fact, if you have evidence that radiation therapy can provide a 90% or better recovery rate and management of the disease, then go for it, okay? But when you take someone with a glioblastoma or some of these other major advanced cancers and you start nuking them, I said that is fundamentally unethical.
Yeah, I got you. You talked about the standard of care, I guess, well, it wouldn't be the surgery, but it'd be the chemo and the radiation accelerating, I believe, metastases or just, the cancer itself in general. What, under the model that you understand, what's happening with the standard of care to drive cancer's progression?
Yeah, that's a great point. Yeah, what happens is when you create, the radiation creates an incredibly diverse microenvironment rich in glucose and glutamine. We published a major paper in Lancet Oncology, one of the top cancer journals in the world, clearly outlining how radiation contributes to the rapid recurrence and death of the patients. So you surgically debulk the tumor, you create a wonderful wound in the brain, and the wound frees up large amounts of glutamine in that microenvironment. And then oftentimes you give high-dose steroids to reduce the inflammation, both from the surgery and the radiation, because when you're irradiated, your tissues and brain swell. So you give high-dose steroids, which elevate blood sugar levels. Can you believe this? So the very fuels you're trying to cut off to the surviving tumor cells are elevated by the standard of care. And then they wonder, the tumor recurred and you died. Well, I mean, you did the very things you were not supposed to do when you understand the biology of the problem. So you don't want to do that. And furthermore, what is known is that when you irradiate tissues, you facilitate fusion hybridization between the cells. So the macrophages that are coming in to try to correct the damage in the microenvironment fuse with some of the remaining tumor cells, and they become the most highly invasive cells that you can possibly deal with. And they'll spread outside of that location, spread through the brain, or even if they're a pancreas, they'll spread through your body, or lung, they'll spread through your body. And it becomes a brush fire you can't put out any longer. So the very
what do you mean they fuse? Like what happens with the macrophage? They fuse actually with cells? Really?
Yes.
What's that called?
Fusogenic processes, a membrane fusogenesis. Macrophages are highly fusogenic cells, and that they evolved to do that to facilitate wound healing. So you get these multinucleated giant cells, you get all these fused hybrids. Macrophages do that to enhance their capabilities of repairing wounds. This is a well-known biological phenomenon. But when you have cancer, that's the last thing you want to do is create a microenvironment that leads to fusogenicity, especially between macrophages and neoplastic tumor cells. And therefore, the cytoplasm of the macrophage gets diluted with the cytoplasm of the neoplastic cell, and they become rogue macrophages, respecting no longer biological order. And they spread through your body, and they survive in the circulation because they are part of the immune system, and they're highly immunosuppressive. So all this crap that you're throwing at them from immunotherapies, that's not going to stop these guys for the most part, and patients are going to die. So you got to know the biology of the problem you're dealing with. If you don't know the biology of the problem, man, you're going to be just, doing all kinds of crazy stuff that's going to be harmful to the patient.
Yeah, I've heard that tumors are very heterogeneous, and then, once you hit them with chemo and radiation, they become even more heterogeneous. But
yeah.
Again, under your model, Is there heterogeneity in their use of oxphos, their fermentation ability, their fermentation pathways, or is it they're all the same within a given tumor?
Yeah, well, they're all the same. Every tumor is fermenting in that population, whether it's a mesenchymal part of the tumor, whether it's a stem cell part of the tumor, whether stem cells can't metastasize, but they grow like crazy. Whereas the mesenchymal kinds of cells, those are the dangerous ones. Those are the ones that will spread through the tissue and potentially leave and enter the circulation, intravasation, extravasation, all these terms that are underlying the metastatic cascade. So the bottom line is that is you want to try to prevent all that. And here's the situation. So they say, well, these tumor cells are so resistant to chemo. Oh, are they? Yeah. Well, why do you think they're resistant to chemo? Because they're from the very P-glycoprotein. It's the name of a protein on the surface of cells that's designed to pump drugs and harmful products outside of the cells. And the energy to run that pump, that P-glycoprotein pump, is driven by fermentation, like energy through fermentation. Can you believe this? So the very chemical you're trying to do to kill the tumor cell is being pumped out because the cells are fermenting. So how do you kill tumor cells? You stop their fermentation. It's not that, I tell you, it's actually, I feel embarrassed to say how non-complicated this whole situation is. So when you see all these people dying from all this stuff, it's because of the absurdity of what they're treating these poor folks with. I mean, once you understand the biology, what I'm telling you not only makes sense, it's shocking that anyone would ever try that in the first place.
Are you able to observe someone's tumor longitudinally, for instance, as they're going through your protocol? What happens? Does the tumor shrink in a different way? What happens to the heterogeneity? Like what parts are affected first and next? And what clues does that give you to, the efficacy of the method?
Yeah. So we have the paper we're working on right now actually addresses that, because the guy was diagnosed with glioblastoma, and he chose no radiation, no chemo, no surgery either. So just metabolic therapy, which was lowering his blood sugar and elevating his blood ketone levels. And the tumor grew very, very slowly. It was diagnosed as a GBM from biopsy at the original thing. It grew very, very slowly. 3 years later, 3 years down the road, the tumor is now becoming slowly larger. It was originally diagnosed as non-operable because of its tentacular appearance from the cells from the biopsy material and from the MRI. So they called it a non-operable glioma. Yeah, we can get some, but we can't get it all. The same story you hear over and over again. But after 3 years, they looked at the tumor and said, this might be operable now. So he made a decision to go in and decided to get the tumor debulked. So he debulked the tumor, chose no radiation, no chemo, and went back on metabolic therapy. And that was 4 years ago. And MRI's analysis are showing residual evidence of something, but it has no malignant transformation and no continual growth. And the guy's out almost 6.5 years with something that would have killed him in like 20 months. So the answer to your question is yes, we can see this. And we've seen it ourselves. And we published a paper in a journal showing how the behavior of the tumor cells dramatically changes under a microscopic examination. They don't invade anymore. They start dying and they stop invading, right? And you don't disturb the microenvironment. What more? I mean, this is like, okay, what more do you want?
Well, what happens if there are existing metastases and someone goes on your protocol? Like, what's observed with the primary versus the metastases? Does one go first? Does communication between them seem to stop? Like, what's observed?
What's observed is that they go away. The spots, the images on PET scan or MRI, and the physicians are often miffed. Wow, what happened? What did you do? Whatever you're doing, keep doing it. It's not like, what the hell's going on here? When you start targeting glucose and glutamine, the damn cells can't live. They're going to go away. They're going to die. So that's what happens. The spots that you had indicative of metastatic lesions go away. All right. And how do you feel? I feel good. The person says, I feel good.
I totally understand that the end goal obviously is living and health and all that. It's just, in science, I'm curious, and I'm sure a lot of scientists are curious about mechanism and what's observed and all that. That's why I'm asking you these nuances.
I don't blame. So the mechanism is very simple. The tumor cell cannot grow without availability of glucose and glutamine. So any therapy that would disrupt the availability of glucose and glutamine to those cells will kill them. They die.
Yeah, I just wonder if there's a preferential killing of, I don't know, maybe the shape of the tumor changes in such a way that gives you a clue.
Well, we've looked at this, and it is kind of striking. There's a whole field of cell death in the field of biology. Cells die from many different mechanisms. There's apoptosis, there's necrosis, death, and then there's whole levels of different kinds of names that they give for these different forms of death. And when we looked at the effect of these metabolic therapies on different kinds of tumors, it seemed like the appearance of the cell as it was dying differed from one kind of a tumor to the other. So I think it has to do with the biology of the cell. And I think that's my best, it looks like mitotic, there's another one called mitotic catastrophe. When we killed the metastatic cells, they died by necrotic death. But when we killed the proliferating stem cells, they seem to die by mitotic catastrophe. I know I have a lot of colleagues in the field that are loving different mechanisms of cell death. Personally, between you and me, I don't give a rat's ass how the cells are dying, as long as they're dying.
What is mitotic catastrophe, by the way?
That's a term used when the cells can no longer form mitoses, which is the lining up of the chromosomes on the metastatic on the metaphase plate. The separation of the chromosomes. They just seem to croak, but they look differently under the microscope. So it's really kind of interesting. They look like fried eggs. My friends get like, oh wow, they get all excited about this. I just move on. I'm moving on to kill them all. I don't really care how they're dying. I think once we get rid of the tumors, we got nothing else to do. Let's go back and figure out how we kill these guys.
Yeah, no, I understand what you mean. I understand. So how many different people have gone through the protocol that you've come up with?
Well, we're building the protocols all the time. We have clinics all over the world that are very excited about this. And, the bottom line is that they're all groping, say, what do we do? I wrote this big paper with my colleagues, the Press Pulse strategy for managing cancer. And, you bring the body into a new metabolic state first. And then, in other words, you bring yourself into a healthy state. You can't believe how many cancer patients are screwed up so many ways in being unhealthy. Some of them have type 2 diabetes, high blood pressure, this, that, all these other kinds of things. And, you got to bring the body back into a state of metabolic homeostasis. And then once you get into a situation where your glucose is down, your ketones are up, what we call therapeutic ketosis, then we use pulsing, like you said at the very beginning, very accurately. How do you take away glutamine, which is such an important amino acid? Well, we pulse. We hit them strategically just for a short period of time. We don't just keep the glutamine hammer on them all the time because the normal cells are going to get hammered as well. So you got to know. And also what we've published, a beautiful paper, showing that once the individual is in therapeutic ketosis, the glutamine drugs can be reduced by half and they penetrate into the tumor through blood-brain barriers 3 times faster than if you didn't do that. So the whole, I'm telling you, it's unbelievable. So you got to know what to do. And this is the cutting edge of where we are right now. If people want to say, well, how come you're not doing this in all these clinics and everything else? Well, we're starting to do it. People are responding really well, but we have not yet, with the exception of a few people, and I don't want to say we haven't because there's a lot of people with triple negative breast cancer, they should have been dead, that are out years later and they're doing very well. It's just that I got to write these papers up. You think it's easy writing up case reports? It's not easy. So, but I write them up. And once we get enough of them, you're going to have a tipping point because people basically want to live. I mean, you're diagnosed with a glioblastoma, man. You're finished. You're diagnosed with triple negative breast cancer, metastasis to lung and brain. I mean, this is not good. And I'm saying we can save these people. We can actually save these people from rapid death. I'm not saying we can cure them, but I can certainly say we can give them a lot longer quality of life. And maybe in the future, we can resolve this completely.
Do you have doctors on staff, or does the person have their own doctor and you work with the doctor?
I work with physicians, yes, okay. In all different countries, all over the world. And we were starting a global society for metabolic. Believe me, there's a tremendous interest on the part of physicians all over the world. I mean, they're in the front lines treating these folks, and they want to see patients get healthy. That's their job. That's why they took on this profession. And when they see the potential of what I'm talking about, and they've actually recognized this in a couple of their patients, they want to know more. And the problem we have is that a lot of the physicians that would like to do this are not trained to understand what's going on yet. They can be quick learners, of course, and they need to be able to do it. And right now, the system the way it's set up, does not allow them to do that. They'll lose their license to practice medicine if they implement therapies that is not sanctioned by the American Medical Association. So they want to do it, but they're inhibited from doing it. And then there's another whole group of physicians that would never want to do it because it might interfere their salary. So you got to look at all, and the hospitals are in the business of generating revenue. And you're going to bring on a therapy that may not generate the same level of revenue, but that can be addressed. I think these things are secondary. I think the bottom line is, can you keep terminal cancer patients alive 2, 3, 4 times longer than they were predicted to survive? And that's the goal.
So how do people that speak to you, how do they get in? I mean, I can see the dietary stuff's under their control, but how do they get these glutamine inhibiting drugs? Where do they get them from?
Yeah, well, that's an issue. And not only glutamine inhibiting drugs. And this is another thing. Sometimes the industry, the pharmaceutical industry or whatever industry, will scarelli these. You know what that means? Yeah, you know what that term means?
What is the term again?
Scarelli. They scarelli the drugs.
Scarelli. Oh, it sounds like someone's last name that they did it 2 years ago.
Yeah, remember Martin Scarelli, the guy who jacked up the price of the EpiPens?
Oh, okay, yeah.
Remember that guy, Martin Scarelli, that low life? All right, so these people need it for, anaphylaxis and things like this. The pens were $3 or $4 a piece. He jacked them up to $800. And he did it because he could. He wanted to make a lot of money on it. So you have various drugs that are sitting around, not thought to be very, very valuable. All of a sudden, you put it in the right context and you see, whoa, this drug is really powerful. So what, instead of like embendazole, right? Canada is 89 cents a tablet. In Turkey, it's like $1 a tablet. In the United States, it's $385 a tablet.
Whoa.
So, I mean, this is the unethical, actually immoral behavior. So, I mean, if you want to manage cancer, you can do it without all the interference from individuals that are driven by greed. So, our job is to manage the disease non-toxically, cost-effectively, and we can do that. It can be done.
Yeah. What kind of cancers have you worked with people on, and have you noticed any differences in survival or progression, et cetera?
Yeah. We've done every kind of a cancer, blood cancers. We've done with people. Now I don't publish this. You say I can't believe it unless it's published. Well, of course. But the ones we're tackling, glioblastoma. I mean, if we can manage that, you can manage almost anything because they're all the same. I look at the metabolism of glioblastoma. It's the same of bladder cancer, non-small lung cancer, triple negative breast cancer. All the cancers the same.
Well, what if they're all the same? What makes some cancers? I mean, why would cancer spread preferentially to secondary sites and not other sites? And why are some aggressive and not aggressive? Like under the models you understand, what drives that?
Yeah, well, that's the macrophage. The macrophage homes to the tissues that it does biologically, and it's a non-random. In fact, it was called seed soil by Paget back in the 1800s with breast cancer. Breast goes to lung and liver. A lot of tumors go to liver. Eventually, they go to brain. And you say, whoa, what could explain that? It was called the seed soil. Well, we defined what the nature of the seed soil is. So there was some experiments in mice where they took macrophages out of mice and labeled them, and then injected them back into other mice just to see where they would go. And they preferably went to lung and liver, the same places. You look at the tissues that have a lot of turnover, like lung tissue, for example, is constantly confronted by potential pathogens from the air that we breathe. So lung has a very high turnover of macrophages. The liver is another organ that's detoxifying stuff in our bloodstreams. So they have a very heavy need for the availability of local macrophages. So when you get lung cancer or lung cancer or these other cancers, they spread to these very organs that oftentimes have a need for macrophage function. So we've looked into all of these kinds of things, and we can come back and parse out, what's going on. And we know once you understand what they need to survive, which is glucose and glutamine, and then you have to say to yourself, well, geez, if that's so clear, how come nobody's doing that? And why nobody's doing that? Why nobody in a clinical trial anywhere on the planet is simultaneously targeting glucose and glutamine like we just published in our major iScience paper? Why is that? If I just told you the key to understanding how to manage cancer, why in the hell is nobody doing that? You should ask people about that.
What would it take for you to be able to do a clinical trial? Is it just money, or are there other showstoppers?
Well, first of all, I'm in a biology department at Boston. We don't even have a medical school, right? So we don't do any of that. But my colleagues in other countries that are in medical clinics are doing modifications of what I just said. I think we can use some of the chemo drugs. They can be used probably at half or 1/3 the dosages that we're giving to patients. When the patient is in therapeutic ketosis, these chemo drugs work a hell of a lot better. Longo's group out there in California has been showing that water-only fasting, you give patients chemo and it works a hell of a lot better, and the patients don't get nearly as sick. So, this is all part and parcel of maintaining a healthy microenvironment in the body and making getting more bang for your therapeutic buck when you do this. You don't give patients with cancer big chocolate cakes and candy like so many people do.
They do in the infusion centers. I know a family member that was in there last year, and that's what they gave her. So,
Oh, well, that's absurd.
I know.
That's absurd. That's almost like criminal medical. It's almost like medical negligence.
I know.
So this is what I call the lack of knowledge. The lack of knowledge in the oncology field is so profound, which contributes to the tremendous suffering and death that all these poor folks are dealing with, primarily from a failure. What I just told you is supported by all these scientific publications. It's not like this is what, you just have to read the literature and understand it and then implement it.
So what help would, if you were able to ask for help right now, what help would you want?
Well, what we do here at our research program at Boston College runs on the good donations of people, right? The good intentions of people. I give them information. I don't charge anything for the information. Travis Christofferson's Foundation for Metabolic Cancer Therapies supports our research massively. And we get money from private foundations and individuals. We've had some success, and these individuals are alive because of what we've done. So we survive. So if people want to donate to our research, they donate to the Travis Christofferson's Foundation. He wrote the book, Tripping Over the Truth.
Yeah, I know that book. Yep.
Which is outlining why the metabolic theory of cancer is exploding right now, because it's the right way to go. When you look in the clinics, most of the therapies are based on the somatic mutation theory of cancer, which we now undermined. So if your therapies, all cancer patients should ask their physicians, their oncologists, what kind of treatment are you giving me? Is it based on the metabolic theory of cancer or the somatic mutation theory of cancer? And it's embarrassing that some physicians don't even know the difference.
Sure, they get it right. The what?
Yeah, right. Well, you can't have that. These guys, their lives are at stake here. Your very soul to exist on the planet is at stake, and you're asking someone who's clueless about the very nature of the disease that he's treating. There's a problem here. There's a problem here. So if the oncologist can articulate in a very clear way why he thinks the strategy that he's using might in fact be better or complementary to metabolic therapy, at least you're talking to somebody that understands the situation, rather than saying, "Oh, there's no evidence for this," or "No, if it were important, I would have heard about it." Well, how many scientific articles do you read? The argument here is a lack of knowledge. Now, our research at Boston College is developing the very diet drug combinations that will eventually lead to the resolution of cancer. And I work with clinicians all over the world, and we adapt these therapies that we develop, because these physicians, they don't want to treat a patient that could harm that patient. If I've done the troubleshooting and done all the different connections to know that this is not going to kill your patient, and your patient's probably going to survive a lot longer, they're very comforted to know that information. So it gives them a little bit of security and understanding that I've tested this and it looks like it works. And the subjects that we're treating are preclinical models that are the best. We've developed clearly the models that best represent human cancer, all different forms of human cancer. And I can tell you that these subjects are healthy, they're responding well, and they live far longer. And I think if we tweak the system for the human, we can get the similar or even better results. As a matter of fact, humans that we've tested do far better than the mice that we're working with. I'm shocked sometimes. I can't believe how long and healthy some of these people, how healthy they are, how long they live. I said, I can't get that degree of survival in the preclinical system. But at least the preclinical system tells us where to go.
Yeah. Have you characterized whether this therapy you're talking about works better early on or later stage, or it doesn't matter? What have you observed?
Well, we've observed every scenario like you've just mentioned, because some people start early on this, and some people, Unfortunately, there are people who come to us with stage 4. They failed every treatment strategy. They're like a walking skeleton. They look like Tammy Baker, Jim and Tammy Baker. Did you ever see her?
Yeah, she was.
It was horrific.
Her makeup and
it was what she looked like from traditional cancer therapies.
Oh, I didn't realize that happened to her. Huh.
Oh God, go on the web and look at it. It's horrific.
Really?
So you can't take a person in that stage and say, okay, we're going to do it, restrict metabolic therapy on you. They won't be able to tolerate that.
Yeah.
So you have to take, and many cancer patients look really healthy, and then they come back from the physician with a long face. Oh, I was just diagnosed with an advanced cancer. Can you believe this? And then they start irradiating and poisoning these guys, and the next thing you know, they look horrible, and many of them die. So if you get them off the bed, listen, the guy that we're working with chose not to do any of that, and his quality of life is very good. He never suffered this. And what we see sometimes is the people who go on metabolic therapy, they have a very high quality of life right up until a couple of days before they pass. And this is very different than standard of care, where they're brutalized and poisoned and irradiated and treated with all kinds of stuff. And they die a slow, painful, excruciating death and debilitating. So, we've a fair degree of success, but we don't have complete success because sometimes the patients, they come and they're already beaten up so bad by the system and they just can't do metabolic therapy.
What about, everyone's attempts for early diagnosis, liquid biopsies, et cetera? What's your opinions there?
Yeah, well, the biopsy, what we discourage biopsies mainly because we have so many articles showing how taking biopsy tissue actually creates an inflammatory microenvironment that could potentially lead to fusion hybridization and spreading the tumor.
Like I had with thyroid, they took this cannula and poked it into my neck to get into some of the areas and to determine if it was cancer. So what could that have done? Would it have driven cancer cells into like a wound tract and gotten them to proliferate?
Yes, that's exactly right. And it doesn't happen to everybody, but it happens to enough. There's enough people where it happens, where there's several scientific reports for all kinds of cancers: brain cancer, lung cancer, colon cancer, prostate cancer, liver cancer. So biopsies, you run the risk of spreading the cancer with a biopsy. So what do you do then? Well, we have plenty of non-invasive ways to know whether or not you have a tumor. So if you go on metabolic therapy and the tumor shrinks really small, you can debulk it completely. Or if it goes away, you don't need to do anything. So why put the patient at risk by doing all these nonsensical things? And then one of the drivers to validate or to say we need to do this is to look at gene expression profiles in the tissue that you biopsy. Oh, you have this mutation, that it's all irrelevant. The mutations don't mean anything. They're all downstream epiphenomenon. If I pull the plug on the glucose and glutamine, the cell dies. So the mutations in that cell become entirely irrelevant. So why? So there's big companies that make $1 billion on doing gene profiles on biopsy material. They're down here in Cambridge, Massachusetts, and they're all over the place. They do. Oh, we got to look at your gene expression profile. No, you don't. Yep. Why are you doing that? Oh, we have to know because we can treat you. So in other words, I got this mutation in my cancer. How am I? Well, we're going to give you radiation and chemo. What the hell? Oh no, we have a special immunotherapy that's going to go in there. But if I pull the plug on the glucose and glutamine, I kill the tumor cell. You don't have to do any of that. So it eliminates that whole field of oncology. You don't need to do any of that stuff. Now, of course, what I'm telling you is like, oh, this is you can't believe this guy. He must be nuts. No, I'm not nuts. The people doing it are the nuts.
One last question. You said that, people will have a longer quality of life with your protocol, but you also mentioned that some of them will just suddenly die. Why would that happen?
Well, because some of them may have taken standard of care. And the other thing, too, is to be honest with you, many of these people that I spoke, we haven't really targeted their glutamine yet. We're just hammering the glucose right now. But once we get on top of the glutamine issue with this cocktail, as I said, it's a diet drug cocktail. There's no food. People always say, well, I got to restrict glutamine. You can't do that. Glutamine is everywhere. It can be synthesized from other amino acids. So you got to use a drug, but you got to know how to wield it properly. I mean, even the guy who does radiation, right? He's not sitting there irradiating somebody's head until the smoke comes out of the guy's ears. I mean, he obviously knows what to do, right? So
hopefully he's not microwaving their head, essentially, right?
Yeah, well, that's what I'm saying. I mean, these guys, they go to school to know how not to treat you so smoke doesn't come out of your ears. But if they want smoke to come out of your ears, you get some numbskull doing it, smoke will come out of your ears and your nostrils and whatever. So, but I'm saying, you got to know how to wield the power of the medication, the medicine that you're using. And you have to know how that medicine works in different environments. So under therapeutic ketosis, you can reduce significantly the dosage and the timing at which you use these medications, which is really wonderful because you can keep the patient out of the hospital from other collateral damage. So many cancer patients suffer in their lives from secondary effects of the treatments they use to cure them. Digestive issues, hormonal imbalances, neuropsychiatric problems. I mean, a person of cancer today, the cancer could be cured, but now you've opened up a whole new array of medical conditions that individual never had, but the fact he was treated with all these toxic treatments. So, we've looked at every nook and cranny of this problem and studied it and evaluated it and know how to avoid these things. And I think it's just a matter of time. I don't know how long it's going to take for the field to come to realize that what we're saying. But the bottom line is that the cancer patient wants to live. And there's a lot of motivation in that.
Yeah. Oh, yeah. Well, very good. Tom, what's the best way for people to reach you if, God forbid, they are a family member of someone they know has cancer and they're interested in finding out more from you?
I have an email at Boston College. People email me. I give them a kit of information. I notify them about Miriam Kalamian and her book, Keto for Cancer. She's a wonderful person, walks a lot of the cancer patients through the minefield and how to do all this stuff, and other resources, scientific publications to support, at least it's a way to give them knowledge so they don't go into these situations blindfolded. And as I said, we get our funding from private foundations and the goodwill of individuals that support us. And we're working to develop a standardized drug diet protocol that will eventually replace the majority of procedures that exist currently today.
Okay. Well, very good. Well, Tom, thanks for coming on the podcast. I really appreciate it.
Okay. Thank you very much for having me.